when we started in the early 80s, we were trying to purify the virus by a sucrose gradient.
Retrovirus particles are purified using a laboratory procedure developed over 40 years ago known as density gradient centrifugation. The cell culture that is undertaken by the scientist to produce workable quantities of virus results in a liquid suspension made up of cells, macroscopic and microscopic cellular debris, virus particles if any are present, and culture fluids. This suspension is spun in a low-speed centrifuge which creates a sediment consisting of the cells and heavier solid material, and, above the sediment, a liquid supernatant containing the much lighter microscopic material. If retroviral particles are present, this is where they will be distributed. Next, a small portion of the supernatant is removed and very gently placed on top of a solution of sucrose. This sucrose solution is prepared in a special way such that its density increases gradually from the top to the bottom of the tube. In this diagram, the layers of different densities are shown as discrete bands, but in the real world of the laboratory, these layers gradually merge into one another. Purification by this technique relies on the fact that particulate matter in the supernatant sample will gradually sink down through the sucrose solution until it reaches a place in the gradient where the sucrose solution and the particulate matter have the same density. When an object gets to this portion of the gradient, it cannot go any further. It is exactly like trying to force a tennis ball to stay put at the bottom of a bucket of water. As soon as you let it go, it bounces back up to where it wants to float according to its density in water. This means that in the sucrose density gradient, all objects of the same density will eventually congregate at the same place in the gradient. In the case of retrovirus particles, this is where the density of the sucrose reaches 1.16 grams per mil. Because the particulate matter in the culture supernatant is so light and tiny, the passage of the sample through the gradient has to be speeded up by spinning the tube in another kind of centrifuge known as an ultracentrifuge. This machine rotates the tube at speeds between 40 to 60,000 revolutions per minute and produces a force many thousands of times gravity. In this diagram, we have assumed the supernatant sample contains retrovirus particles, and you can see them gradually moving through the gradient and being arrested at the 1.16 grams per mil density. Here is a short demonstration to illustrate how a sucrose density gradient solution can be used to separate objects of different densities, in this case beans and macadamia nuts. In this simplified version, we will use a two-band density gradient consisting of water with a density of 1 gram per mil and sucrose at a density of approximately 1.5 grams per mil. First, we prepare our sucrose solution by dissolving sucrose, ordinary table sugar, in water. Then we make our two-density layer solution by carefully pouring our lower density layer, the water, on top of the denser sucrose layer. Next, we place our sample on the top of the two-layer solution. Since these objects are so much larger and heavier than virus particles, gravity is more than sufficient to propel them through the gradient. In fact, as you can see, in this experiment the separation is virtually instant. There is no need to spin the glass in a centrifuge. The result is one density band consisting of nothing but beans, and another density band consisting of nothing but macadamias. In other words, these objects have been successfully purified. What may not be obvious is the part your eyes play in this experiment. Without looking at the two density bands, which is equivalent to performing the electron microscopy, you will not be able to tell if any objects are present, what is their morphology, and whether or not they are pure.
Well, what's the purpose of the purification then?
Well, to make sure you have a real virus,
So Montagnier claimed to prove that he had particles, he had proteins which are not present in any other retrovirus. He purified the virus. But he did not publish any electron micrographs of the purified material to prove, you seeing is believing. And they claimed that they had purified material, but there were no electron micrographs. And this is one of the conditions where Barré-Sinoussi and Jean-Claude Charmaine put it in 1973. You must have picture of the purified virus and show that the material contains nothing else but particles with the same physical characteristics. But no such pictures were published.
Is it important to photograph where the virus is banned in the gradient?
Yeah, because retrovirology has also some history. These are established techniques. If you go to C-type particles, they can be easily bended and they are stable. And if you think HIV is a new retrovirus, you have to go the same way, just to be acknowledged as a retrovirologist dealing with that new virus in a proper way.
Hans Gelderblum, he said it's important to photograph the density gradient where virus is banned. Why is that an important step?
Because this is, electron microscopy, for example, it's always very important to see the morphology of the virus particle. It's in the region of the density gradient, okay, where you think you have a virus purified, and I will say in good shape. If you check after by electron microscopy, the electron microscopy will tell you, okay, you have the right structure of the virus particle with all the envelope which is on the surface of the membrane, okay. Because very often when you make purification, you alter all this process, making sucrose gradients and centrifugation. It's forces, on the virus, pressure on the viruses, and it does not like it too much, especially for HIV, where the envelope protein, which are anchored on the membrane, are not covalently anchored. So that means that very easily you can lose the envelope protein.
Oh, the GP120?
Yeah. So it's important to check then on the electron microscope whether the structure of your virus is the one that you are looking for, with all its viral protein, the envelope protein, structural protein, everything. You have the right shape, the right structure.
And it also shows you what isn't there, correct?
Exactly, also.
So you're saying Montagnier never proved the existence of a new retrovirus because he didn't photograph in the test tube?
Or, as I said, he says it is essential to purify the virus to prove that it has proteins which are not present in any other virus. That's the only way to prove that you have a new virus. But he did not publish pictures. So since he did not publish pictures, we don't know what he had in his purified virus. It is possible that he had purified virus, but it's possible that he did not have anything there. And that's what we've been asking from the very beginning. Why there were no pictures, which are essential to prove purification.
When purifying, Gelderblum told me it's important to photograph the density gradient where virus is banned. Why is that a crucial step?
Well, Gelderblum, I know him well. He's a good electron microscopist in Berlin. And actually, he gave me the best picture of my virus.
Oh, really?
Yes. Well, of course, in order to purify, you have to make this sucrose gradient density equilibrium to have a sharp band. And if you take that sharp band, you have almost pure. Not completely pure, because there are cell vesicles which have the same density. This is why you don't see in the picture, you have a mixture of cellular vesicles, and viral particles.
So, is it really important, or is it not really important?
For us, it's not important. But some people say, if you don't have complete purification, how do you know the disease is not caused by something else?
To silence them, How come you guys didn't just show pictures from the gradient instead of just the culture?
We first show it from the culture. Just by centrifugation, but not synchronous gradient, just by making a pellet of the virus, you could look at it also this way. But here, of course, you have many impurities, coming from the cell. The synchronous gradient has the advantage to partly purify the virus, but again, even in the band of the virus, you have also cellular vesicles, which have the same density, but not the same look, of course, at the electron microscope.
And you're saying that in the purified banding area, there can be other contaminants, and that's why pictures are essential?
Yes, in the purified, the method they used, you can get, by this method, you can get material which is not viral, but it has proteins, it's cellular fragments. The cellular fragments can, with this method he's using, could be at the same place. And then you can have there only cellular fragments, or you can have a mixture of cellular fragments and viruses. But it's important, it's crucial to have a picture.
If you separate by density 1.15 gram per ml, you have a lot of vesicular stuff inside, not related to virus. Detritus, a reaction product of the cell. You have a lot of host cell constituents in that band. That's not too nice.
So are there other particles that can look like retroviruses in that band?
Yes. In that material you can have cellular fragments, and they have proteins and they have RNA, which retroviruses have, and in fact they may even look like retrovirus particles. So it is important to have, it is crucial to have an electron micrograph of the material for us, for example, and for any other scientists to believe what they are claiming.
When you purify HIV, there are some challenges because it's contaminated with cellular debris.
As I said.
And particles that look like retroviruses but are not infected.
Yes, as I said.
How do you distinguish between what is infective and what isn't?
You cannot.
Montagnier gives crucial importance to this band, because he said if the particles do not bend at the 1.16 gram per mil band, then they are not retrovirus particles.